Hello and welcome to my slideshow in computational biology background, specifically in genetic variants. So, as you know, there exists the central dogma of biology, which dictates how DNA becomes RNA becomes protein. That's like the basic path that most people commonly know. However, inside real organisms, inside real cells, the central dogma is just a small piece. It cannot fully explain the transcription translation pipeline from DNA to protein. It's not just as simple as you look at the DNA and you can just get the protein out. And the differences in the genetic code will significantly complicate this. Like, oh, there's a mutation somewhere in the DNA, it will significantly cause complications. So further on, I'll be referring to these as variants, which are changes in the genetic code of DNA that may or may not result in phenotypic differences, and the differences may be beneficial or detrimental. So basically, they can be pretty much anything, technically. And these variants, though, we have to classify them, because as is human nature, classify things. So there's a classification system. This is the classification system, which ranges from pathogenic to benign. And the middle here is uncertain significance. So as you can see, if it's above 90%, it's likely pathogenic. Above 99%, it's pathogenic. If it's below 10%, it's likely benign. And if it's below 0.1%, it's benign. So these classifications, in the clinical sense, help prevent overreaction, which means when thinking something is a bigger problem than it actually is. So like, oh, I think this is a very big problem, but it's actually just benign, or likely benign. And false assurance, which is the opposite, which is when thinking a small problem is, thinking something is a small problem than it actually is, which basically means I think it's not going to be a problem, I think it's like uncertain or likely benign, but it's actually likely pathogenic or pathogenic. Notice the classifications are defined. You have to be above 90% or less than 10%. So all this like middle area here is uncertain significance. And it's important to also note that there's these percentages, something called posterior probability. You may notice that it's classified with posterior probability. If you know what that already is with the Bayes formula, this is slightly different. It's used by a framework called the Tavtigian Bayesian Framework, which was established by Dr. Tavtigian in 2018. And it basically established a quantitative metric for qualitative measurements. So what that means is that originally it's based on quality, but that can now be defined with numbers, so quantitative. It's an abstraction of the old ACMG or AMP strength of evidence system, which basically is a sort of dictation of how strong the evidence is to classify it in any direction into a simpler points-based system. So you'll see what that is. The Tavtigian system assigns evidence basically based on the strength of that evidence. So, like, let's say you have something that supports it being benign and strongly supports that would count as -4 points. Or something that supports as pathogenic, but is not very, like, it's supporting evidence, not very significant, then that would classify as 1 point. So all of that is then added up and then placed on this line over here to basically, these points, basically determine where it is. So, like, let's say you have 5 points in total, then you would be uncertain significance. And if you had like -2, you'd be likely benign. So here's the background to odds and probability. This is why the Tavtigian system works. And the system relies on the conversion between odds and probability. So basically, odds are will occur divided by will not occur, while probability is will occur over total. So like odds for rolling a dice and getting a 6, well, will occur, it's 1, 6, and will not occur can be 1 to 5. So it's 1 over 5. Well, probabilities will occur over total. One instance it will occur, but the total is 6, so it'd be 1 over 6. And odds can be from 0 to infinity, while probability is confined to 0 to 1. Because, like, let's say you're looking for anything except 6, then this will be 5, this will be 1. But for probability, this will be 5 and this will be 6. And the conversion for them is pretty simple, as you can see here. Odds is equal to probability, 1 minus probability, and probability is equal to odds, 1 plus odds. So it's rather simple there. Here's the actual formula for the Bayesian framework. And I'll explain where the 0.1 and 2.08 come from. You just create posterior odds, and to get posterior probability, just use the previous one, like this one here. Posterior odds 1 plus posterior odds. So why does this framework work? Because the way logarithms work, you can basically make multiplication act as addition. So, natural log of 3 plus natural log of 4 equals natural log of 3 times 4. This is important since combining odds is done with multiplication. So finding the posterior probability involves multiplying the prior, as saw in the equation, multiplying the prior odds, which is 0.1, with an exponent of 2.08 to raise the number of points. The prior odds tend to be 0.1, 1 to 9 odds, and the reason we specifically chose 0.1 and 2.08 was because it aligned with the classifications already done before the Tavtigian system was implemented. Because actually it was implemented pretty recently, considering it's like only a couple years ago, like not that long ago. So it's supposed to like match up with the old framework that they use for backwards compatibility. So you don't have to like reclassify a bunch of things, a bunch of variants. So you can see like this applies here 2.08. This matches with negative 1 point, well positive 1 point. This matches with negative 1 point. You see that? So for instance, say that you have two pieces of evidence that support for a certain variant. One of them is PVS, which means pathogenic very strong, that counts for 8 points. And the other one is PM, pathogenic moderate, which counts for 2 points. So you add the two up, they count for 10 points in total. So using the prior equations, 0.1 times 2.08 to the 10th equals 151.577, which is your posterior odds. And turn out the probability, you get 0.993, which is basically 99%, like, sure, 99%. So, since it's greater than 0.99, it can be classified as pathogenic, as you can see here for the posterior probability, which is sufficient evidence for a pathogenic classification. And as you can see here, above 10 points means it can be classified as pathogenic. So now that we've successfully classified the data with the Tavtigian system, the data needs to be stored somewhere. So there's this database called ClinVar, which is a data source provided by the National Library of Medicine that stores all of the information about the variants. So they store large amounts of variants and their classification. So like, is it pathogenic, is it benign, is it uncertain significance? And most of the data for this classification originally was mostly sourced from patient cases and like population cases, which is in vivo information, which means in life, which are cases in living organisms, or tests in living organisms. So there is a problem with this though, with in vivo data, because in vivo data isn't very, like, you can't get a lot of it, nor can you get it easily. So do the necessary 90% and 10% brackets for classification for likely pathogenic or benign, many variants can only be classified as uncertain significance. And uncertain significance, you don't really know what that is. Like, it's uncertain. You can guess, but it's uncertain, you're not sure. And this can cause overreaction and false assurance in the clinical field, which is bad. And the problem being is that we don't have enough data to classify certain variants as pathogenic or benign, basically. So how do we fix this problem? The problem is that we don't have enough data and don't have data on specific variants in particular. So we want to examine the effects of multiple specific variants. To do that, we can run labs to test the effect of changes to the DNA sequence to get the amount of data. And this is through a process called MAVE, or multiplexed assays of variant effect, which determine the effect of change in the DNA sequence, or variants in the DNA sequence, on both coding and non-coding regions. I'll explain what the rest of that means, but multiplex basically means that all the variants are put inside the same cultures, they're being separate, so it's not like there's one test tube per variant. They're all in a big, like, test tube, sort of. Disclaimer. MAVE is called in vitro testing, which it means it's not done in the living organism. It translates to in glass, probably refers to test tubes or other testing things. And these in vitro like analyses in lab basically is not a substitute to in vivo in life cases. They're just a point for possible pathogenic variants. So say for example like a MAVE test uncovers a variant that is pathogenic, then in vivo cases that would be closely monitored, basically. So this is a type of MAVE which involves changes in the coding regions. Do recall that MAVE can involve changes in both the coding and non-coding regions. This is how it's done in the coding regions. So coding regions, as you know, actively change the protein structure, which changes its function. So the way this is done, the entire process, basically the first step is to create large amounts of slightly different genomic sequences with single nucleotide variants, or SNVs. And that basically means only one nucleotide in the entire sequence is changed. You'll see why that's done soon. And these are put into cells via plasmid or CRISPR, you'll see that soon, and are transcribed into proteins because the cells do that. After that, you assess the status of the protein with a variety of methods, such as adding a drug or marked antibody, or just a variety of other purposes. And you'll see here, you can you can find the differences in the proteins with this method. So, for example, look at this here. The effectiveness of a protein by providing drug resistance can be tested with selective growth, this method here. So, if a certain variant in the DNA is enough to produce the protein, then the drug will kill it because it lacks the protein to protect it. But if the variant creates a hypofunctional protein, which is a protein that functions but is bad, basically functions less well, then some of the variant could survive, but less. And if it works fine, then a lot of the variant would survive. So that would change the ratios, because the starting ratios are known. But after that, it could be like, oh, these die out because they cannot survive. And this one can kind of survive, so it doesn't make that much, and this one survives very well, so it makes a lot. And then these ratios can be recorded in the presence of by the presence of a certain variant after the stressor is introduced. So let's say the stressor is adding the drug, then these are gone, so you can know that their protein was probably non-functional. So there are two types, like subgroups, I guess, of the coding region modifications, which are called DMS and SGE. Basically, the DMS stands for deep mutation scan, and involves, after you make the variants, involves adding them to bacterial cells via plasmid. And the differences between DMS and SGE is SGE, stands for saturated genome editing, involves using CRISPR, Cas9, to insert the variants into mammalian cell cultures instead of bacteria. And this is the more preferred and commonly used method because for the modification to coding regions, due to the fact that you use mammalian cell cultures instead of bacterial ones, as you know, bacteria don't have that, like, the intron-extron cutting, so it more closely resembles human cells compared to the bacterial plasmids. So I did talk about coding regions. Now there's another process called MAVE. Oh no, MAVE includes both coding and non-coding regions. And just talk about coding regions. Now this is MPRA, or massively parallel reporter assays, which is the analysis of non-coding regions. So non-coding regions, basically for this test, they determine the change on how much RNA is produced. So when you start the process with the variants, you have the whole list of variants, and then you start modifying. You attach a DNA barcode to the coding area to basically mark it, and when it gets transcribed to RNA, you can figure out what's being transcribed, and then you also add the variant to your place of interest. And these are then added to cell cultures, and the production of RNA is examined, so how much RNA is produced. And these are used to assess the change in transcription due to the variant, determine the regulatory strength of the variant. So let's say I add a variant to a non-coding region, and that significantly drops the production of the DNA barcoded RNA. I can know that variant will significantly reduce how much protein of that coding region we produced, basically, because there's a lot less RNA. And all of this also creates data. So data from MAVE labs, which includes DMS, SGE, and MPRA, are stored on a publicly available database called MAVEDB, which is a database that basically stores the effects of all these tests. And it's basically used for finding the variants that possibly have been confirmed pathogenic in vitro in the labs by the MAVE test, and these possibly dangerous subjects are subject to closer monitoring to see what they do. Now, there's a problem with MAVE testing as well. Now, remember when I was talking about the coding regions, I said that MAVE labs commonly use SNVs, or single nucleotide variants, meaning only one nucleotide in the sequence is changed. This is fine, like, we've been doing this is fine. say a nucleotide sequence is 100 nucleotides long, if you do every single possible single nucleotide variant, there can only be 300 variants, since you can change any of the 100 spots on the 100 nucleotide sequence, and you can change any of them to the other 3 possible nucleotides. So let's say it originally was T, then it could become A, G, or C. That's fine, there's only 300 variants, that's very possible. But let's say we want to change 2 nucleotides on the sequence. So that means you gotta change 2 points. So 100 times 99, because 100 is any one of the 100 nucleotides, and we already counted one, so any of the 99 remaining nucleotides. And since they change independently, you have to account for all 3 variants. So it could be TA, T could become any of the other 3, and the A could also become any of the other 3. So that means 3 times 3. And if you multiply all that together, it equals 44,550 possible combinations, which is very large, significantly larger than the 300, and this is not fine. This is called a combinatorial explosion, which basically means the combinations go up very, very quickly. So basically the numbers are too big for labs in vitro testing to handle. So, computers. Computers can handle very large numbers. They are good at doing that. So basically computers were used for this to solve this combinatorial explosion problem. In 1987, they coined the term in silico, which basically means test done with computers, as reference to computer chips being made of silicon. And since the process has been around for a while, but in recent times, advancements in the field have been significant thanks to AI. As you know, AI is good at this. AI can do this. And this can be used for a pointer for wet lab testing, such as the in vitro MAVE labs. It's kind of like a pointing pipeline where in silico does a very broad scan, finds any suspicious stuff, and that's used for in vitro MAVE labs, which finds possibly pathogenic variants, and that is then used for in vivo testing, or in vivo data and classification. So that's pretty much the background for computational biology and why it's important and how it helps, what role does it play in the whole genetic variants, the whole area. And my next presentation will be on the recent advancements in AI and how they play a role in silico testing.